I'm sure many of you have been in this situation. Academia and science as a whole remains inaccessible to many. How are you supposed to keep up to date with the latest research if papers are behind paywalls, if conferences cost hundreds, and most don't have access to elite institutions? I can't get you accepted into Harvard, and if I just started sharing journal PDFs, my head would be on a pike. But I can bring you something better: reports from the front lines of science, talks, presentations, and ideas from the most forward-thinking scientists in the field. And what better place to start than the largest mycology conference in the United States, the Mycological Society of America's annual meeting. This year it was held in Madison, Wisconsin. A few friends and I made the trek north by car, and once there I assembled a crack team of scientists, and also me, to bring you a front row seat to the cutting edge of mycology. My name is Clancy here at CraterAlias, and what happens at this conference? It's better than any paper. So, let's get into it. I'll address the elephant in the room. Scientific conferences are also expensive, from registration to room cost to travel costs to food. For many scientists, these expenses can be covered by grants or by their respective universities. However, this isn't as readily applicable for undergraduates or even graduate students who cannot rely on these sources. However, the Mycological Society of America, MSA, provides travel awards and student awards, a smidge over 20, alongside Spores, the official mentorship program, which sponsors another 20 undergraduates, fully funding their trip. I did neither of these. Private funding is an option for many, and now is a good time to talk about who funded my trip, the Boston Mycological Club. It's one of the most well-established mycology groups in the country, and it's the oldest continually operating amateur group, being established in 1895. Alongside monthly meetings, regular forays, and professional lectures, the BMC offers research grants and community outreach grants, alongside a scholarship to attend national forays hosted by the North American Mycological Association. I applied to and received a community outreach grant to go to MSA, which covered the vast majority of expenses. I cannot thank the BMC enough for their support, and you'll be hearing about them in this video as well as a few more to come. If you're in the Boston area, I cannot highlight them enough. But on to the first day of the conference, which I was not there for. There was only one event, the Hsueh Camp, hosted by the International Hsueh Consortium. The inaugural in-person meeting of the consortium, they all focus on one genus of fungi, Hsueh. They're ectomycorrhizal and mostly associate with pine, but they're great lab pets, which makes them super interesting as a candidate for a model genus, something which ectomycorrhizal study mainly lacks aside from a few select species. Model species and genera are nice to have. They allow us to make more general assumptions about related groups of fungi. Now, these assumptions might not be true, but they're more likely to be true, which means it's easier to argue that you should get funding to study them. Genetic transformation on Hsueh allows you to dive deeper into the individual proteins and pathways involved in symbiosis, and as a pretty large genus, it can also be used for evolutionary study or systematic study. Hsueh is just one genus of fungus that all fall under the ectomycorrhizal lifestyle, but there are still such high potential in this group, corroborated by the dozens of scientists around the US that hosted the event and the dozens more that attended. You can keep up to date with their research on their website, and don't be surprised when you hear about this genus very, very soon. I got into Madison at like 10 p.m., so after a quick shower, I went straight to bed in the dorm accommodations. Day 2. After one of the nicest student dining experiences of my life, I went to go attend one of the optional events held throughout the week. This one, however, it would be remiss of me to not mention, the MSA Annual Foray. It's where all the mushroom nerds, me included, go out into a forest and try to find fungi. This year it was held at Black Hawk Ridge. A short drive away from University of Wisconsin-Madison, it hosts oak, pine, plains, all perfect for finding fungi. With 150 people in attendance and a large wait list to boot, it was quite the event. If you've ever been to a large foray event, it's a lot like that, but instead of like one mushroom nerd, there's 100. It was a pretty normal foray, aside from the sheer scale of it. What made it interesting was the amount of specialists that joined the group. Not everyone was looking for mushrooms. Some were digging for truffles, taking a hand lens to any discolored leaf. Thus, the final haul was massive and super interesting. After an impromptu bus interview, I grabbed my badge, washed off my sweat, and rushed to attend one of the many special meetings held throughout the event. Special meetings cover specific topics like IP and mycology, or how to use free Joint Genome Institute tools, or they cater to specific groups. Case in point, the Spores meeting. It was a meet and greet for the mentors, mentees, and coordinators of the program. I'll be talking about them a lot more in a future video about mentorship in mycology, so be on the lookout for that. The student and postdoc section business meeting was right after. It introduced the new officers, covered what happened in the last year, and goals for the next. I attended this one because I was contractually obligated to. The final event of the day was the MSA reception. Free food, free Wisconsin beer, and undoubtedly the room with the most mycologists in it on Earth. But that'll happen a lot this week. It's really an event to meet people. Anyone could walk up to anyone else and strike up a fungal conversation, network with potential advisors, or make general connections with others in the field. Looking back on the event, it matched the overall tone of MSA. People with decades of experience, more than happy to hear about your research and openly engage. Also free food. I was running around trying to introduce myself to people I've been emailing over the past few months to set up interviews. Once again, stay subscribed for that. But as the hors d'oeuvres reserves dwindled, day 2 came to a close. Day 3 was really the first true day of the conference. Posters, talks, and more, kicked off by the presidential address by who else? Dr. Timothy James, MSA President. It's up to the current president regarding the subject of the talk, but it's usually a mix of research and personal stories behind that research. It's also customary that the president receive no questions after the marathon that is an hour-long talk. The name of the talk, The Quest for Origins in Fungi, is a bit misleading. The Tim Talk gave insight into his life and experience as a student, researcher, and eventually principal investigator. The kind of stuff you wouldn't find in any of Tim's papers, a peek behind the curtain. His focus is on early diverging lineages of fungi, the true zoosporic fungi, namely Allomyces and Rosella. He touched on challenges in his research, challenging genomes, lifestyles, as well as the moving and upkeep of the University of Michigan's fungal herbarium. The presidential address is, of course, to everyone. However, a majority of the audience is in academia, so this year, more than any year, a specific topic had to be touched on. The funding cuts imposed by the current administration are felt in all areas of science, but particularly fungi. MSA has done their part, lifting registration costs for many attendees, but obviously it can't support everyone. Many rely on grants from the Department of Agriculture, Department of Energy, or the National Science Foundation, and funding freezes trickle down. People are scared that they have to freeze work at their lab or stop accepting students. In this, there can be a lot of anger, but we need to stay focused on the future. Tim's message was that we need to band together as scientists and as communities. We ought to inform to uplift. Tim didn't take up all of his time. In keeping with the meeting's theme, cultivating mycology and mycologists, he had to invite a specific group up to the stage. A new initiative started this year, the Sclerotia Project. Following in the footsteps of Ben Franklin, the idea is to save up a bunch of money, put it in a vault, and let it sit for 100 years. In the meantime, letting it invest and compound.
We probably can't change today's situation. There's not a lot we can do to change tomorrow's situation, but we might be able to do something about the situation 100 years from now if we play our cards right. So,
They want this project to be yearly, which requires more students to help save for the future.
we're trying not to put too many names or faces to the project so that anyone can take it over.
But it's the message that stuck with me and many others.
It's kind of this tiny vote we can cast saying that, yeah, we believe that mycology should and will persist hundreds of years into the future.
If you want to donate to a future mycologist, the link is hopefully up there, but if not, it'll definitely be in the description. Speaking of those future mycologists, many of today's were presenting in one of two forms, either a poster or a presentation. With over 300 presenters total, you can see why I had to assemble my crack team. Talks were split up over 3 days, and there were still 4 talks happening at the same time. Talks were grouped by somewhat topic, some were quite similar, but others narrowed down the scope. The sessions were held close enough to each other that you could go in between them, though some session rooms were, let's say, more suitable to host these sessions. It was really just this one, not enough chairs, unfortunately. This did not take away from the talks, of course, which were either 15-minute full talks or 8-minute flash talks. I want to go over a few that stood out to me and my crack team as either particularly interesting and or applicable to those outside of academia. One issue that I've had my eye on that's been undercovered, at least scientifically, is invasive golden oyster mushrooms, Pleurotus citrinopiliatus. Ayeshwara Veerabahu, a PhD student at UW-Madison, is really the first person to study their invasion extensively. They give a talk on it here at MSA pre-publication, though now it's been published in the Current Biology Journal. We know golden oysters are invasive. First appearing in New York in 2013, it likely actually escaped a few years earlier in 2010. From there, it spread to Wisconsin, the rest of the northern US, into Canada, and now it's spreading to the south. Golden oysters are saprotrophs. They eat dead things, mainly wood. Golden oyster mushrooms take over these decaying communities and push out native species. Aishwarya's work aimed to better understand the actual effect golden oysters are having. She sampled dead tree stumps that had and didn't have golden oyster mushrooms, and then analyzed the species present therein. Stumps with golden oyster had significantly lower diversity and altered community structure. Aishwarya also utilized climate data to try and figure out where golden oysters might be spreading next, and yeah, we should be worried. You can help out with her project by using the apps you already do.
If you find golden oysters, take a picture. A lot of times your phone, if you take the picture with your phone, it will automatically record like geolocation data. And so just have an iNaturalist account or make one and then upload that observation, and it would be extraordinarily helpful.
You can contribute directly by sending information at the link, hopefully up there, but definitely down in the description. AI is another big topic this year, with increasing reliance on LLMs like ChatGPT to help identify mushrooms. We're in this sort of weird spot where genetic techniques have way surpassed the normal field identification, meaning that species are now other species, or things that we thought were species are now species complexes. This makes identification impossible for the average forager. AI and iNaturalist are undoubtedly powerful tools, but are we cheating ourselves by using them? The primary talk on this topic was by Dr. Emily Cantonwine, touching on how they use AI in their science curriculum. She works with intro biology students, many of which are only taking the class just to fulfill a gen ed. The traditional "you're only cheating yourself" approach doesn't really work with these people because they're not going to be identifying plants or mushrooms in the future. So how do you make these people not turn to AI as the solution? Make it part of the process. You check your ID with the info from something like iNaturalist. Then you iterate, make a hypothesis. Does this make sense? Consult with guidebooks, online. Make AI a tool, not the authority. We've all seen those Google searches. Onto some TLDR talks, because if I covered them all, this would be a 20-hour video. Editing Clancy here. This was actually a 40-minute long video initially. Okay, that's all. Back to the video. On to surprisingly material science with Dr. Deborah Lynn Porter, who touched on the uses of fungi in engineering. Dr. Porter emphasized how physical and mechanical characterization actually scales up. This is helpful for understanding pathogenic fungi like armillaria, as well as general hyphal structure, useful for biotemplating. Porter's focuses on this biotemplating process and how this deformation happens through geometric analysis. Heather Dawson gave a talk on truffle conservation with her dog. It wasn't on stage, but dogs are used for truffle hunting, though usually it's your choice edibles, your magnatum, your melanosporum. However, dogs can be used to find all truffles, and Heather Dawson thought, why don't we do that? It's less destructive than just raking around in the dirt, and dogs are better at finding truffles than we are. So she uses it to help better catalog this ecology and diversity of these underground fungi. Justin Egan had a super catchy title, Addition by Subtraction, regarding a specific strain of Penicillium expansum. He found that a reduction in expression of primary metabolites actually increased the diversity of secondary metabolism, particularly one peptide. While the peptide was interesting, it's the mechanism that I found really cool. Like, imagine if you stopped eating and suddenly you just became like immune to arsenic. Like, that's not what happened, but imagine. Jason from earlier had a talk on spore shape. Certain Amanita spores can be grouped into particularly fat or particularly thin groups, and these confer properties such as having more nutrients or being able to travel farther. This variance in spore shape actually allows for multiple spore dispersal strategies in a single fungus. I can't emphasize how tip of the iceberg this is. There were fungi for agriculture, rewilding, effects of climate change on mycorrhizal communities, plant communities, endophyte communities, plant pathogens, animal pathogens, systematics, phylogenetics, metabolomics, ocean fungi, fire fungi, and most important of all, Basidiobulus. Yeah, baby, Basidiobulus. I've been informed that it's actually Basidiobulus. Oopsies. The day finished with a poster session, the first of two. They put all the presenters in the room, 100 total, and they present their work on You'll never guess it, posters. I actually gave one of these, but forgot to take a photo in the moment because it was super chaotic. Posters are distinct from presentations in that they generally covered smaller projects. For instance, side projects, which are worth it to make a poster, but not the time, effort, or stress of a presentation. Others were people early in their study. Maybe they didn't have data yet and were looking for some feedback on their work or hypothesis. Many applied established tools to answer simple yet necessary questions about fungi, something that I greatly admire. As the saying goes, we stand on the shoulders of giants, and also grad students. It took place in one big room, and with 100 posters, 99 of which I was not an expert on, there is no world in which I could cover them all. So I chose someone at random who definitely isn't my friend. Alex's work is on the zombie or entomopathogenic fungi. Alex looked at a few major ones and found that all but one had very small genomes. We actually expect this from parasites, but why is a question for another day. What is interesting here is how big Mycosporids' genome is. While she continues to study this, I asked her about her experience at MSA.
I love presenting at MSA. I didn't get to do it last year, the first time I was here, but this year was really exciting. I got a lot of questions. People always love zombie fungi, so thankfully I have an interesting topic to study. But it was really good. People are really communicative, and I have so many questions for anything that you do.
That's the great part. And I guess there's so many like great mycologists here, so many people who are experts, super niche experts. How is it like talking to some of those people?
It's like weirdly an honor sometimes, and you're like, oh my God, I know you, and you're coming up to my poster. I've heard
cited
so
you in a
much about
paper,
you.
right?
Exactly. And then also some people who are like experts in a totally different field will come up to you and have such good insights, even though they don't study anything that you're like remotely related to. So that's also really fun.
Great.
Day 3 ended post-poster session and with a dinner at Madison's very own Old Fashioned. Whoa, day 4 already? It commenced with the second of 2 keynote talks, the Carling Lecture. The talk was named after Dr. John Sidney Carling, and if you have a few minutes, you should read this man's biography. He had a truly fascinating life. The Carling Lecture highlights the work of a researcher not an MSA to help showcase the wealth of diversity in mycology. The invited speakers come from around the globe, and this year was no exception, with Dr. Yen-Ping Hsueh, a Taiwanese scientist who hosts a lab at the Max Planck Institute for Biology in Germany. She was invited to talk on her area of expertise, carnivorous fungi. Now, mushrooms aren't known for their speed, but they can still hunt. Dr. Hsueh's talk was on nematode carnivory. Nematodes are these like microscopic worms that live in soil and a bunch of other places. You might know them from SpongeBob. The main 2 structures we see in these nematode-eating fungi are adhesive knots and constricting rings. Now, these adhesive knots are little nubs with sticky stuff on the end of them that trap nematodes. The nematode sticks to it and can't leave, and the fungus eventually takes over the stressed and starving nematode. Constricting rings are similar, and instead of a chemical trap, it's a mechanical one. If a nematode crawls through one, the trap tightens, and the nematode can't escape, eventually leading to the fungus taking over it. The final one Dr. Hsueh studies is the neurotoxin defense of oyster fungi. Oyster mushrooms produce little drops of neurotoxin if they detect nematodes, and if a nematode touches the drop, it dies and becomes easy prey for the fungus. TLDR, glue trap, snap trap, Walmart radioactive shrimp trap. Dr. Hsueh gave some background into her previous work, and then dived deep into the exact proteins and mechanisms involved in this relationship. Dr. Hsueh showed some of her videos from YouTube where she plays her own music over nematode v fungi action. If you want to delve into the nitty-gritty, her papers are also on her website, but I expect I'll be talking about nematode carnivory sooner rather than later. I knew a bit about carnivorous fungi before, but the breadth of knowledge that Dr. Hsueh and many others showcased this week was captivating. Following a quick refreshment break, the next session started with symposia. Symposia are kind of like normal talks, but they had to be submitted way in advance, about 6 months instead of 3. And they also had to have a cohesive theme with multiple speakers, which were usually more experienced. PhD candidates, professors, postdocs, niche experts, and the like. There were only 6 of these symposia, 2 a day held at the same time. They were super insightful in their respective topics, and I'll give a cursory overview of all of them. The first pair was Future Fungaria and Mobile Genetic Elements. Fungaria, or fungal herbaria, hold dried specimens of fungi to be used as a species reference or as historical or geographical data. Herbaria, plant and fungal alike, are facing reduced funding and, in some cases, closure. The symposia focused on how we should tackle this issue. There were two schools of thought. We should catalog and protect data, and data is for nerds. It wasn't exactly that. It was more focused on community building, that we should get people to appreciate the specimens for more than just pieces of data. I leaned a bit more this way, but having those people that care about your institution as more than just databases is a huge boon for herbaria. Mobile genetic elements covered pieces of the genome which are more likely to move within a genome or into another genome of another species. In many cases, they're common in fungi, and may be the source of large shifts in evolution, like fungi gaining new proteins, pathways, and sometimes ways of living. The highlight of the symposium, according to my buddies, was the final talk on fungal starships. Starships are these large pieces of genetic material which move from fungus to fungus, particularly in pathogenic fungi. This makes many of them super adaptable and thus hard to deal with in the field or to treat in a human. They're also found in domesticated strains of Penicillium and Aspergillus used for food production. Looking forward to hearing more about this. It's super cool. The next pair was fungal bacterial frenemies and underground explorers. Fungal bacterial frenemies covered fungus-bacteria interactions, both cooperative and competitive. There are so many examples of this happening, but I feel the symposium did a good job covering a wide array of techniques such as metagenomics, carbon tracking, and stable isotope probing, as well as application in Basidia bullus, effects in fungal molecules called lactones on bacteria, and how we can apply this knowledge to plant-fungi-bacterial symbioses. Underground Explorers was led by SPUN, the Society for the Protection of Underground Networks, namely Dr. Adriana Corrales. The symposium focused on the effects that SPUN has and how it can positively influence underrepresented communities. A majority of the talks came from students who were partially or fully funded by SPUN. Looking at endangered Hawaiian nanu trees, Honduran agricultural land, coastal gopher land, and Midwestern drought, interesting case studies all in their own right, but if you're interested in soil or underground mycorrhizal conservation, SPUN is the premier organization. Final pair, we'll speedrun these, were mycological education and fungal futures. Mycology education was super interesting to me. It's kind of what I'm doing right now, believe it or not. They touched on interesting ways to teach, undergraduate mentorship, the 4DEE framework, and finding ways to engage outside of the classroom at museums and 4-H. They also touched on online tools like ZygoLife and iNaturalist, and Dr. Corrales' interesting experience searching for fungi in the understudied, fungally uncharted rainforests of Colombia. Finally, fungal futures. How are fungal metabolites affected by climate change? This includes toxins for human and plant pathogens, as well as metabolites for common soil species like penicillin, and how these changes may imply unforeseen effects to humans and agriculture, plus how we can solve it. The short answer is keep an eye on it. MSA isn't just for established scientists, but I haven't really touched on the sheer scale. The vast majority of talks were from students, undergraduates, and graduates. As such, the MSA aimed to facilitate inclusion and student involvement. So started the student and postdoc section, SPS in
2013.
The SPS still hosts many events, including a student lunch and trivia night/mixer to help foster student connection. The SPS also hosted a new event, Mycologists and Musings, where it's a Q&A with just a single mycologist alongside a complimentary breakfast. This year they hosted two professors, Dr. Joseph Spadafora and Dr. Lotus Lofgren. They touched on their experiences being department head and opening a new lab, respectively, as well as a bit on their work, history, and consulting for The Last of Us. Wait, what? While these are just two of many dozens of professors who attend these events, it inspires students to go out and interact with those who maybe they didn't have the confidence to interact with before. This would unfortunately not be helpful at the next event, the MSA Business Meeting and Student Awards Breakfast. While us students and various other attendees enjoyed our free breakfast, various speakers came up, from the MSA President, Treasurer, and Spores Representative. Money stuff, new MSA initiatives, this year the DEI chapter, Spores, and where the next few meetings will be held. 2026 will be in Portland, Oregon, but what we didn't know is that 2027 will be held in the South/Southeast. Humidity enjoyers everywhere rejoiced. They also gave out the student awards. No surprises here, they announced them well before the meeting, but congrats to everyone who got one. After another round of symposia, lunch, and presentations, I had to leave. I had an 8-hour drive ahead of me, and I wanted to get there at a reasonable time. The events I ended up missing were the MSA banquet, awards session, and auction. Throughout the week, people have dropped off their mycological items to be put up for silent and live auction, where the proceeds go to MSA. The banquet and awards ceremony are the send-off to the event. The banquet is a super nice dinner, and anyone who has the time to attend does. They also hold the live portion of the auction and hand out the non-student awards. Most of the awards were known beforehand, being listed in the program and online for things like Distinguished Mycologist Awards and MSA Fellows. But a few of the awards were impromptu, including student awards for best presentations, including 2 of my crack team! Yeah! Best undergraduate talk and best graduate poster. Congrats to them. And a full list of the awards is online on MSA's website. And that's a wrap. Just about everything that happens in the largest mycology conference in the US. Mycology is always growing and will continue to be pushed forward. The MSA annual meeting was such a blast that I'll actually be producing at least 3 more videos on the topic and related things on mentorship in mycology, education in mycology, and more. So again, like and subscribe to keep up to date with those. I'll be going to MSA 2026 in Portland, and I hope to see you there. If you have any questions about mycology or any topics in mycology, nitty-gritty about conferences, anything, let me know by dropping that in the comments. And in the meantime, continue to mind your mycelium.